Hello, my name is Sangmyung Lee, and I'm a rising sophomore at the Vanderbilt University. I am currently intern at the Indiana University Cyber Infrastructure for the Network Science Center in the Dr. Katie Boner's lab. And today, our presentation is comparing the tablet sapiens to the Human Reference Atlas. So the advent of the single-cell resolution analysis and the advance in the bird RNA sequencing enable annotation and the segmentation of the single cell, and the offer opportunity to explore the molecular characterization of the cell types and the spatial location of the single cells. This technology have enabled detailed comparison at the cellular level of the vast amount of data and make possible to creation of the cellular atlas of the larger tissue block, entire anatomical structure, and soon complete organ, and one day, the entire human body. The Human Reference Atlas Common Coordinate Framework captured the anatomical structures, cell types, and the biomarker to represent the relationship among the cells in the different tissue and the associated 3D reference object. The Human Biomolecular Atlas program is developing, validating, and optimizing various technology for discovering the human body at the single-cell resolution. So, our research question is, how does the HRA compare to the experimental data, such as the tablet sapiens data? In the study present here, we demonstrate how the anatomical structure, cell types, and the biomarker table can be used to showcase contribution by the tablet sapiens dataset for tracking progress toward the inaccurate complete ASCT plus B table. The tablet sapiens dataset is a single-cell transcriptomic atlas that provides the integrated molecular reference of around the 500,000 cells and the 45 tissue blocks from the 24 organs across the 15 donors. The anatomical structure, cell types, and the biomarker table is the framework that captures the ontological descriptors and the spatial reference object. Tablet sapiens data was downloaded from the cell by gene portal. First, we mapped the anatomical structure, names of the 45 tissue blocks to the 24 organs covered in the tablet sapiens. Next, the 24 tablet sapiens organs were compared to the existing 26 ASCT plus B master table to identify the set of the organs for which ASCT plus B table and the tablet sapiens data exist. Cell ontology ID and the Uber anatomy ontology are specific neural ontology covering all major cell types and the anatomy. The ASCT plus B reporter let users explore the anatomical structure, cell types, and the biomarker, their interrelationship, and their linkage to the existing ontology and the supporting literature. To do this, we first unified the names of the corrected cell types for the identical cell types by removing the cell types mapped to the same name in the cell ontology ID. Sex, ethnicity, and the development stage for all 45 tissue blocks were used to cluster sample into 15 donors and perform cell count analysis. Tissue blocks and the cell counts for each organ from the tablet sapiens for each donor were counted and visualized using the Excel on the tablet. Afterward, only the segmented organ tissue, they observed the difference in the cell count between the two groups was corrected by the additionally calculating the variability between the cell by gene and the extracted data. A comparison of the organ data from the tablet sapiens and the ASCT plus B table was performed. We used the two main criteria to classify tablet sapiens tissue block into organs. First, the publicly available raw data from the cell by gene portal for each organ. And second, the supplemented material which included tissue procurement information for each organ. As a result, 24 tablet sapiens organs, among them, there are 16 shared organs, 10 organs only in the ASCT plus B table, and the 8 only in the tablet sapiens as the Venn diagram shown. As the vasculature data reported in the tablet sapiens covered both the blood vasculature and the lymph vasculature in the ASCT plus B table, it was kept separate. The ASCT plus B reporter was used to compute the difference in the anatomical structure, cell types, and the biomarker between the tablet sapiens and the ASCT plus B table data. The ASCT plus B reporter makes it possible to compare the tablet sapiens data visually to existing ASCT plus B master table and the reported discrepancies that future ASCT plus B table revision might be addressed. The total of the 115 shared cell types reported and the 211 unique cell types were from the tablet sapiens were reported. The small intestine and the uterus and the ASCT plus B table did not have a cell ontology ID provided, so the identical cell type matching to the tablet sapiens could not be performed. We also observed that some organs, such as the blood, pancreas, and the prostate, share more identical cell types, but they were identified as a new cell types, and different level of the cell ontology classes driven them. Interestingly, cell annotation of the tabula sapiens in the pancreas is expressed differentially by the cell annotation tool. Tabula sapiens generated cell annotation for by all organs by automated annotation tool called as popular vote, while the Azimuth, the web application that uses the automated annotation reference dataset, generate the different set of the cell types in the pancreas. This result provide insight into the future need for the unified protocol and the cell annotation tool for the cell type comparison. The tabula sapiens provide a total of the 483,182 cells from the 24 different organs across the 15 donors. Among the all donors, donor 14 had the highest number of the cells, and the 69% of all cells come from the 4 donors. This is unique as many studies origin collected from the multiple donors with the different biological goals, experimental procedure, and the computational approaches, which make it hard to replicate it and or compare the result. However, the tabula sapien data was carefully constructed to include multiple organs for 4 donors and making it possible to control the genetic background, age, environment exposure, and the epigenetic effects. This study compared the unique experimental data set with the evolving HRA and report the result accuracy and the coverage of the HRA. The 8 new organ and the 211 unique cell types from the tabula sapiens dataset can be used to see future ASCT plus B table. Experimental data can be used to see new ASCT plus B table that do not yet exist in the HRA and to identify organ experts that might be like to serve as the HRA authors or reviewers. Also, we observed that the cell ontology was expressed differentially by the cell annotation tool. Not only non-identical cell ontology ideas or differentially identified cell types require the additional evaluation to distinguish them. This result suggests that significant amount of the comparative data may have been missing from the study. As such, comparing the anatomical structures and the cell types across different atlas for the experimental data set reveal the complexity of the comparing result from the different body location, donors, and the experimental procedures, while which highlighting the need for the unified reference framework. However, comparing tabula sapiens or other high-quality data still provide valuable insight to the evolving HRA, make it easier to determine how to improve the ASCT plus B table. Lastly, I would like to give a big special thank you for the NIH and the HuBMAP for the opportunity here to the Dr. Katie Bonner's lab, and also thank for my faculty mentor, Olin Kordorkis, and the Kylie Bonner, and the graduate student, Vicky Daya, for helping me a lot and teaching me so much for this summer about the data visualization and the data analysis. I am happy to take any question now, and please don't hesitate to contact me by the email address listed above if you have any other question. Thank you so much.